Okay, before we start out, there's something fairly interesting. If you look at, say, the human genome, and this is typical for vertebrate genomes, you see the part that codes for protein, all the 22,000 genes, are only about 1 to 2% of the total DNA in our genome. Now, you might double if you start adding the various, promoters, enhancers, and all this stuff in between. And maybe make it somewhat larger if you throw in all the intron sequences and stuff like that. But when you add all of that stuff together, anything that has anything to do with protein-coding genes, whether you're talking about the intron sequences, the promoters, the enhancers scattered all over the place, all that kind of stuff put together is only about 5% of the genome, or less. There are DNA sequences that don't have any genes in them but play structural roles. For instance, the telomeres, the sequences at the ends of each chromosome, that those don't code for any genes. There are what we call origins of replication. Those of you who had genetics probably heard about replication origins. Prokaryotes generally have only one. Eukaryotes have multiple in each chromosome. Those are binding sites for proteins that assemble together all the complex for DNA replication. Okay, no genes there. You have other things that are solely structural. The kinetochores of a chromosome. The center of that is long DNA sequences that code for no proteins, but they act as assembly sites for proteins that will then bind to the microtubules of the spindle fibers, in other words. So they're important for separating proteins. So we have structural stuff. We have a lot of things like these replicated transposons, those sequences that are repeated hundreds, thousands, even millions of times in the genome. In fact, this RNA interference mechanism may have evolved as a way of keeping these guys in check so they didn't, stick transposon sequences in important genes and things like that. Keep them from running wild, keep them under somewhat control. So we've had all this kind of stuff. And there's still a huge amount of DNA. The question is, what's it there for? Some of it's probably just, sometimes use the term junk DNA. It's just baggage that's carried along and replicated like everything else and doesn't do any particular harm, it doesn't do you any good. It's there for the ride. Okay, that's some of it. But recently, there have been some studies. There's a project called ENCODE. What it means is encyclopedia of, let me get the acronym here.
What page is that?
Oh, what page? 119. Oh. Encyclopedia is something, a DNA elements. The idea behind this is to start looking at genomes, both of humans and other vertebrates, find sequences in common, try to get an idea of what these things are doing, and what's transcribed and what's not. But last year, there were some results published looking at the human genome, and their estimation is that somewhere close to 80% of the genome is transcribed. Previous studies in mice also gave a similar number. Now that's pretty astonishing when you consider protein-coding genes and stuff, everything associated with just a few percent of the genome, structural regions a few percent of the genome, and then, add some more stuff for these transposons and these duplicate sequences and stuff like that. When you look at this, the majority of genes in humans, the majority of DNA in humans and in, at least in mice, presumably many other organisms, is actually transcribed into RNA. We don't know what the heck most of that stuff does. It's a wide open field. What are all these RNAs here for? What do they do? Now, some of these things are also transcribed, what are often called pseudogenes. A pseudogene is basically a copy of a gene that has enough serious mistakes in it that's not going to make a functional protein. Yes?
What is the first letter?
First letter, P. Yeah.
Okay.
Pseudo, fake. Okay.
I wasn't sure.
Oh, all right. Okay, so pseudogenes are like a copy of a regular gene, and maybe it's missing something, or it's got an insertion in it or something. The end result is that it does not make a functional protein. Many of them actually are transcribed, but with all the mistakes in them, they never get, those precursor RNAs never get processed, and they eventually get destroyed. So that's some of it. And we probably have thousands of pseudogenes in our genome that are basically useless. Okay, yeah?
So a pseudogene won't what? Won't make a...
It won't make a functioning protein. It may be missing critical sequence or what have you. And the RNAs for these things, although many of them are indeed transcribed, the RNAs for them aren't properly processed and they get stuck and destroyed in the nucleus. Okay, so that's some of it. But the question is, what's all this other stuff there for? That's suggesting something. That's suggesting, now, obviously synthesizing RNAs takes energy. You have to use nucleotide triphosphates as an energy and a nucleotide source. Tear the phosphates off. It takes effort to make these things. And if you're making tons of RNA, using up all those raw materials and using up all that energy, and it's not doing you a darn bit of good, there's going to be evolutionary pressure to get rid of that. Why? If you only need to transcribe 3 or 5% of your genome and you're transcribing 70%, that's a humongous waste. They put the federal government to shame. Okay. Not a bad analogy, right? Okay. If you can stop transcribing a whole bunch of this stuff, assuming it doesn't have any use, that's going to be a benefit to you because you're wasting less energy, wasting less raw materials. And you can do that, use that stuff you save to put something that really helps your survival. Okay, so it's suggesting the fact that we see this in a wide range of organisms that we've looked at, that most of the genomes being transcribed, it's saying that there must be some functional significance to all that stuff. It must play some kind of roles. Now the big question is, what roles? That's where we have a huge mystery, folks. We have a few ideas. I'm going to put a couple more neat things out about this whole concept. We have a few ideas. We've found that there are various classes of regulatory RNAs, and we're going to look at one class that kind of intriguing. We don't know huge amounts about them yet, except with a few examples. But there must be a whole bunch of other stuff that have functional significance, that are important. And we keep on transcribing these. But the vast majority of these transcribed RNAs, we simply don't know what they are used for. And what that means is the field of RNA biology is poised to explode. There will be enough to keep RNA biologists busy for decades, if not centuries, figuring out what does all this stuff do. We only know a little tiny bit of it. It's like having a jigsaw puzzle that's supposed to make a pretty scene, and you've got 5 pieces out of 500. And the idea now is to find the other 495 pieces and assemble them together and see how things work. This is a really hot field. I mean, what we don't know about gene expression, what we don't know about the roles of these RNAs is tremendous. And it says, hey, there's a lot of stuff we've got to work on. Yeah.
Like, okay, what kind of field would you go into to do that kind of research? Like,
Molecular biology.
I mean, like, what kind of, how would you be employed?
Oh, well, one thing, a lot of it would probably be the research universities. But the pharmaceutical companies, now, drug companies employ an awful lot of cell molecular biologists and biochemists. The problem with the drug companies, of course, they're interested in the bottom line. And especially in the United States, the pressure is that quarterly report. That's what makes you or breaks you on Wall Street. And the problem is so many companies just think, okay, where are we gonna be on the next quarter? And a lot of times the executives get their pay based on what the quarterly reports are like. So, you know, you have, you wanna call it selective pressure for short-term profits and forget the strategic stuff later on. That's why we've lost so many industries to China and Japan and stuff like that, which tended to think, where are we going to be 10 years from now or 5 years from now, not where we're gonna be in the next quarter. I mean, case in point, look at certain personal bias, but look at American cars, okay? They are still not up to the standards of the Japanese cars. They were complaining about, oh my God, the Japanese cars are taking over the market. They actually had the federal government step in and impose what were called, quote, voluntary import restrictions on the Japanese companies in the 80s and stuff. And it was about as voluntary as somebody coming up and pointing a block to your head and you voluntarily give them your wallet. And that's about basically what it was. And, you know, the thing is, the Japanese cars tended to be higher and still are to this day, always driving Mazdas for that reason. They tend to still be put together higher quality than the American equivalents. And these guys in Detroit just don't get it, and the taxpayers have been bailing them out. That's not good. But, yeah, so that's the kind of issue. And that's the thing, the pharmaceutical companies, they're looking for things that are going to often be short-term, you know, that you get short-term results. You're going to get results quick. That's one reason why a lot of them ramped back. For a while, they're plunging into RNA interference research, but the payoff wasn't as immediate as they were hoping. So they ramped their R&D back on that quite a bit, which is unfortunate because it has a lot of potential. But, you know, the potential might not be met for 5 or 10 years, and these guys, meanwhile, are putting out crappy quarterly reports. So that's the kind of thing. So a lot of this is still probably at the level of basic research. And, you know, quite a bit in terms of basic research in this country. Most of it, well, you get some of the foundations like American Cancer Society and that type of stuff. And they put money in some states like Texas put a lot of money into cancer research. You know, other states have put state money into certain types of biomedical research. A lot of it's done through the federal government, National Institutes of Health. They have, I think, maybe 15 billion, nowhere near enough, but maybe 15 or 20 billion dollars a year go into that for individual labs, most of them associated with big universities like UAB. And these folks are laying the groundwork, understanding the basic mechanisms. And then, if something looks hot, then the drug companies jump on it. But the main thing, the basic research is still mostly government and some private foundation kind of things, and some states are involved in it. So that's the kind of, this type of stuff, extremely interesting. Who knows what we're going to come up with? But that's going to be a long time in the future. So that's probably still more academia at this point. Okay, but it is interesting. This is going to be extremely hot field. Okay, so we're seeing huge portions of the genome are being transcribed. Now another thing. People using computer databases and sequences and genomes of a wide range of organisms, invertebrates and vertebrates included, started scanning these things and asking, are there highly super conserved sequences in these genomes? In other words, sequences that are very similar or identical from one organism to the next, even if they're widely divergent organisms, like say an insect and a human. And sure enough, they found hundreds of highly conserved sequences. Now, some of them we already knew what they were. There are certain proteins, and genes for certain proteins like histones are very highly conserved. We mentioned that certain regions in the 28S ribosomal RNA, the region that form the active site of the ribozyme that joins peptide bonds together, those are invariant in everything we've looked at. You change a single nucleotide in either of those two regions and protein synthesis is completely abolished. Okay, so we find some, we know what some of these things are, but they found hundreds of extremely highly conserved sequences in a wide range of different genomes. Most of those sequences were not anywhere near or associated with protein coding genes. These things were scattered throughout the genome. Sometimes you'd find these super conserved sequences inside introns, inside certain introns. Sometimes you'd find them between genes. But you'd find these things, and these things were extremely highly conserved, sometimes almost invariant, even from extremely, lower organisms like protists all the way up to human beings. If you have highly conserved genetic sequences like that, it's telling you those things are critically important for life, and they are intolerant to mutations. Not that these things aren't going to mutate. It's that if you have a mutation in that sequence, you're history, you're toast. So that's saying there's things out there that we don't know what they're doing, but they're absolutely critically essential to life itself. Question is, what the heck are these things? So that kind of introduces us to a very interesting type of thing. There are huge numbers of transcribed RNAs of presumably important, possibly critical function, but we don't know what they are or what they do yet. There are genomic sequences that must be essential for life, for just about all kinds of multicellular life, at least all kinds of animal life, that we don't know what they do yet. The field of RNA biology and genomics is just opening up. Sequencing genomes was just the first baby step. Now, figuring out what all the sequences do and what their functions are and how they interact, or the products of these things interact with each other, that's the next step. So we're a long way from really understanding how life works. Even though, sequencing genomes of vertebrates is actually getting to be relatively easy. I think they can do it for about $5,000 now if you have the right equipment. Oh yeah, the first human genome project, when they first started, the idea, they were predicting it was going to cost $20 billion to sequence the human genome. And it was going to take something like 10 or 15 years. It was done a lot less than that. For a tenth of that, it cost $2 billion. Now, when you have a government-sponsored project that is way under budget and way ahead of time, that's probably the first time in U.S. history that that's ever happened. Well, now you can get a genome sequence for between $5,000 and $10,000, only about 10 years after the first sequences were released. And the hope is, and this should probably be in the case within 5 years or so, that new technology will allow a genome to be sequenced for under $1,000. That means doctor's office, he draws some blood, sends it off to the sequencing lab, and look and say, here's some risk factors. Oh, this drug could have really horrible side effects for you, so we're not going to use it. We'll use something else instead. That type of stuff. So that'll actually be important information when it becomes cheap enough that a doctor will do that on a routine basis, and the insurance companies will pay for it. It'll give us a lot of insights into risk factors, into what drugs may work or what drugs may not, into potential nasty side effects of certain things, whatever. So, technology is getting pretty impressive, but once again, sequencing genomes is just the very beginning. Figuring out what these things do, that's the tough part. Okay, so that's sort of like an introduction to some of this. And what we're going to do now, just tell you this is a hot field here. This is cool stuff.